I think I'm going to go ahead and start. So this talk is a little different. This time I'm going to use slides, but I might write a little on a chalkboard. So this week I presented some background on age and stage structured populations, and then we did a little background on quantitative genetics. And the reason I did that was because I wanted to present some recent results from my lab that sort of use both. And I thought it might be nice to see how you could sort of use both approaches and put it together to ask a question. So my lab, my research group, is really interested in applying evolutionary models to organisms that have complex life histories. Organisms that have more than one form of reproduction, or have, life histories that have intrinsically sort of interesting aspects to it that we think are fun. So, so I feel like you guys have seen, a fitness landscape. This is nice because I didn't draw it, right? This is, Mathematica drew that. Thank you. So, we might imagine some fitness landscape, and we're going to be agnostic as to what's really going on. But and we're showing some sort of, it could be a local maxima or a global maximum, but nevertheless, there's some maximum of fitness at some phenotype, which we'll call, Z bar or something. So we might imagine that at some point, if we look at a population of organisms, that the mean for a trait that has been under adaptive evolution might be at a fitness peak, maybe a local fitness peak, and that that trait could be just one thing, or it could be multivariate, like we talked about this morning. But we might imagine that it's sitting there. But then, sort of, we've already discussed this. We rarely think that that fitness landscapes will be constant, and we might imagine that there's some environmental change, and that environmental change could be anything. And some, if we think about the fitness that maximizes, the trait value that maximizes fitness, it might have a new value, we call it theta. And now the population is sitting at a place of lower fitness. So it's maladapted. So, it depends on how far down it is, and we were sort of talking about being maladaptive, how far you are from the optimum. That was the sort of cue in the last talk. And so what can happen? Well, many things can happen, but one thing that could happen is we could have adaptation sort of to this new peak. So the term evolutionary lag, sometimes it's called lag load, is often defined as the difference between the phenotypic trait mean currently in a population and some optimum. And this is how Maynard Smith defined it. This is a scaled version. Later on in the talk, I'll present an unscaled version that's just the absolute difference between these two things on some value. And we sort of intuitively expect to see greater evolutionary lag when environmental change is fast. So if the environment is changing quickly, we might expect to see more evolutionary lag. If it's changing more slowly, maybe less. So, so one of the things, if we go back to this, when you're at lower fitness, presumably the population is either increasing more slowly or perhaps even decreasing, right? We talked about when lambda is less than 1, population might be decreasing. All right, so if that happens, the question is, what's going to win? The demographic effects of decreasing population number, or the ability of this thing to move up this peak? And this process is sometimes called evolutionary rescue. So this is the world's most simple diagram possible. But if you imagine maybe population size here and time this way, and I love that I've labeled them. So this is when a theoretician makes a graph. Okay? So we have a population with some size, and if we imagine there's an environmental change that moves that population to a place of lower fitness, it's possible that that lower fitness leads to population decline. So now we've got this demographic decline. The numbers of organisms are going down. If there's sufficient adaptive variation, additive genetic variance in the population for whatever this trait is, we might imagine the selection rescues it, and population size goes up. Okay, so that seems nice. Of course, this picture ignores everything that's stochastic about this. So if you imagine that a population has small population size, it's also possible, but it's also possible that the population goes here. Yes, it does have enough variation, but stochasticity happens and boom, it goes to 0 and it goes extinct. So that's a question that people who are interested in sort of eco-evolutionary dynamics ask is sort of what time scales are these happening, and how likely is it that you end up down here versus that you go back up again. So the question that I'm going to talk about today is the ideas of life history and environmental change. So the question we were asking is, how does stage structure and clonal reproduction affect a population's ability to do this tracking of change? Right? So what is going to happen? So I always feel like there should be maybe at some point an organism, even though I don't work on any. So stage structure and clonality. So what kinds of organisms are we interested in? So we're interested in organisms that have multiple forms of reproduction, so both asexual and sexual reproduction. And some of these organisms have the kind of stage structure that we talked about a few lectures ago. So here's Vitis rotundiflora, I think. So this is an invasive, that's on a beach in Hawaii. It doesn't look like a beach in Hawaii. But it's spreading invasively, and this plant spreads by growing vegetative runners. And so probably much of this picture is a single clone. It also can reproduce, I think, sexually. And so, you know, if we were doing a stage-structured model of that, maybe we would use the size of the plant that's connected as some sort of, we're developing our model that we were talking about before. Down here is staghorn coral. And, you know, coral species, they have sexual reproduction, but they also have budding of polyps and fragmentation that can also lead to spread. The other two pictures show different kinds of clonal reproduction. This is everyone's favorite lab rat for this sort of thing. This is Hydra. And what Hydra is doing here is it's having a bout of clonal reproduction. That is a new Hydra that is budding from the parent. And over here, this is Poa, which is a grassy thing. It looks like, these look like the kinds of reproductive seed structures that you might develop through sexual reproduction, but those are actually something that they call bulbules, and they are not produced sexually, they're asexual as well. All right, so, you know, organisms, they exist. So we're going to combine, then, two things that we've been talking about. We're going to combine phenotypic evolution with a stage structure life history. So we're going to put those things together. So we're going to follow the sort of track taken by Barfield et al. And consider a multivariate phenotypic trait, Z, that's made up of some n variates, some arbitrary number of variates. And we're going to take sort of this quantitative genetic approach and say that Z is made up of sort of two components. A component that is the additive genetic component, the genetic component that if you're having random mating and sexual reproduction can be handed on. And this component here, E, which I sort of, it's not just the environmental component, it's all the non-additive genetic as well. There's all that extra stuff that we kept saying, oh, we'll worry about that later. So it would include, you know, things like dominance, things like epistasis, and other stuff like that. So we're going to follow two things in the model, and some of this should be a little, you know, familiar. We're going to follow the number of individuals in each stage, and we know how to do that because we talked about that in a few lectures. We talked about using a transition matrix approach to do that. And we're going to also follow the joint probability density function for genotype and phenotype of each stage. So we imagine that there's a distribution for each stage of the possible phenotypes and genotypes. At the beginning, we're going to be agnostic as to what that distribution looks like, and later we'll make some assumptions. So we're going to develop recursions for these two things. So this is in here because often when I give this talk, I haven't talked about this before, but you guys are all experienced and could probably give the next part. So in a simple age-structured life history, we can describe the life history with a life history graph. And as we did before, of course, my notation is not consistent. When have I had consistent notation the entire week? Not even once. So these were the P's earlier, and these were, I think, M's. You know, we've completely changed it. That's fine. Still, survivor probabilities and fecundities. And here's our friend the Leslie matrix for this very simple case. And, you know, it looks exactly like we did before, so that's nice. But then we can say, well, what about the kinds of organisms that we're interested in? And the kinds of organisms that we're interested in, these are no longer ages, they're stages. And so while you can move to the next stage, you can also stay in the same stage. Because it might be the size, like the size of a cluster of plants, or the size of a coral colony or something else. And also, while you can have, I'll always use, for my models, I always let stage 1 be sexually produced organisms if there's sexual reproduction. So we have, you know, sexual reproduction, but there's also other kinds of transitions. So one of the things that we did is we divided up these transitions into 3 kinds. One that we just called transitions, which is movement through survival, I guess a growth or developmental growth from one stage to the next, some probability of remaining in the same stage. And then we could imagine that these might be shrinkages, right? So you move backwards, you can become smaller. Yes? Yeah, usually herbivory and things like that. You make it smaller. If it's size, yeah. I'll give an example where that doesn't happen, but this is the most general case where it's possible. So if this is size, you can move back. If it's a developmental stage, these would be 0. Yeah, that's a good point. But for all of these kinds of things, getting bigger, staying the same stage, maybe getting smaller again, you might imagine that the organism that starts here and the organism that starts there has the same genotype and the same phenotype. So it takes both with it. That's not what happens with sex, right? With sexual reproduction, the organism that's the parent, or the 2 organisms that are the parent, contribute to the genotype, which presumably also contributes to the phenotype, but not directly. So this is a very different type of transition. And then finally, we're going to imagine some type of clonal reproduction. Later I'll have an explicit one. But maybe stage 2 can also be reached by budding. So it might be a size of a single polyp, and something, maybe that's by budding. And then this is a case where the transition does involve making a new individual, but presumably that new individual has the same genotype. And then we'll talk about what happens with the phenotype. Okay, so we put all that together. And this is just the same thing we've written several times, except that little bar is there to remind me to say that what I'm writing here is the transition matrix. I should have written a big A with a bar, IJ. A big A with a bar. These IJs are transitions in stages, not genotype. And that bar is the average over the full distribution of genotype and phenotype. Okay? So the number of individuals in stage I in the next time unit has all the movements from all the other stages sort of averaged over phenotype and genotype. And we can separate these out into these 3 pieces. And we can separate n into 3 different numbers. That's fine. And then these are these individual transitions. And really, these should have, G, Z, because they'd be specific to a phenotype and a genotype, but sort of being sloppy. We'll see it more carefully later. Okay. So again, we want to study phenotypic evolution. We're following this phenotypic trait Z that has this additive genetic factor, and then E, which I'll just keep calling it environmental factor because I hate saying such a long thing, but you will remember that what I mean is non-additive genetic plus random environmental factor. It has all those things in it. So there's 2 things about clonal reproduction that we wanted to take into account in the model in these idea of when you transition, what do you take with you? And the 2 things are the possibility of somatic mutation. So in organisms that don't sequester their germline, or that produce offspring from somatic tissue, like that hydra, if you have somatic mutations, mutations that occur in the non-meiotic cells, they could be incorporated clonally into the next generation. And that affects the mapping of a parental genotype onto an offspring genotype for clonal offspring. So normally, the whole point of cloning is that your offspring are genetically the same as you. So here we're allowing your offspring to possibly be a little bit different from you. Later on, I'll talk about how we modeled it, but basically what you should know is we assumed that the mutations, somatic mutations, were random with respect to fitness, so there's no sort of directionality to it. And then we also thought about something else, this environmental component of phenotype, E. It includes, again, non-additive and environmental deviation, and we thought it might affect the mapping of how we get phenotype for clonal offspring. So generally, we assume in random mating sexual populations that there's no correlation between E in the parent and, well, E star, I guess, in the parent and E in the offspring, right? That's sort of our assumption of that random model that we developed. That the only thing being passed on was sort of the additive parts. But here we're going to think, well, what if there's some sort of correlation between those 2, just for clonal offspring? We're going to call that rho, this association parameter. It's not quite a correlation unless we make certain assumptions. And let me sort of walk you through the thinking there. So this is this association. So we might think Well, some of it depends on the genetics, right? So it depends on if we set up situations where dominance and epistasis exist. Because if I give my exact clonal genotype to my clone, I give not only my additive, but I give everything. I give so every dominance interaction and every epistatic interaction is preserved. So that might inflate my resemblance to my offspring, at least that part. But you can imagine that there are types of asexual reproduction. These are all over my lawn every year. This is asexual reproduction in dandelion. They're making clonal seed, asexual seed. But apart from that, apart from the fact that there's no meiosis, in many ways it's similar to other members, other plants that are very closely related that produce those same seeds sexually. So they're sort of physiologically sort of similar. But we might imagine rho to be close to 0. 0 is the rho value for sexual offspring and random mating. So if it's similar to sexual reproduction, it might be close to 0. It might be higher than 0 because of what I just said about dominance, deviations, and epistasis. It depends on your model. But rho might be closer to 1 for an organism whose offspring phenotype sort of comes out of a fully developed adult phenotype, if you like. So if there are aspects of the phenotype of this hydra that develop over time, you might imagine that some of the offspring that it clones from its body, so to speak, will have those same, that little clone thing has experienced the same sort of environmental effects that the parent had. So it might be higher. So this is going to depend on the type of clonal reproduction, but in general, we think it will be non-zero. Okay. So it's a discrete time model. Remember we talked about discrete, continuous. This is why I spent so much time with discrete, because this model is discrete. So I was like, fine. So this is the same sort of thing we did before. And then we need to think about the change in the joint probability density function of genotype and phenotype. And what we did is we separated out. So in order to think about the joint distribution of genotype and phenotype in stage I individuals in the next time frame, we think about what proportion of individuals, that's the NI, come from transitions, come from sex, come from clonal reproduction, and then we have sort of these separate functions for the three of them. And I'm not going to go into detail for these two, except to say that for sex we assumed random mating, and yeah, we assumed random mating. And this is just a transition, so it doesn't really. Initially, we are sort of agnostic to what's happening here, but later on, to get the final answer, I'm going to assume a Gaussian distribution of phenotype and a Gaussian distribution of genotype. Now, you should all say, oh, for phenotype, maybe that's fine. We've already talked about how many phenotypes have a Gaussian distribution, at least if you measure it on the right sort of scale, possibly a log scale. But for genotype, is that likely? Under random mating, sexual reproduction, and some assumptions, yes, you can get that. And the question is, will it deviate? I'll talk a little bit later about how we tested to make sure that that assumption wasn't really affecting our model too much. So let me just focus for a little bit on this function kappa. So this is the distribution of genotype and phenotype in stage I individuals that were produced clonally. Look, okay, I circled it. So for that, we have to think a little bit about how do we develop that. So we might assume that we integrate overall genotypes and phenotypes that were in the parent. And this is the PDF, the joint PDF of the parent. So that's the distribution of the parents. This is the transition from I to J. So we're trying to get to I. And this is the joint PDF of a stage I clonal offspring that has a certain genotype and phenotype, whose parent was stage J, and they have some genotype and phenotype. So we have some transitions in this distribution. And we can separate that out into two parts, one for the genotype and one for the phenotype. And in general here, this is the distribution of genotype and here we have the genotype in the parent and the phenotype in the parent. And as long as you assume that there's not transgenerational epigenetic effects, genotype shouldn't depend directly on phenotype, right? It should depend on genotype. I mean, it will indirectly. So we're going to go ahead and drop that and do it like this. Okay. So, so we're going to go through both of those bits. First, the distribution of the genotype and the clonal offspring given the genotype in the parents. For that, we just have to think about somatic mutation a little bit. And so what we assume is just that we have some random deviate M, which is due to mutation, we just assume it's this distribution. And just to give you a sense of what it might look like, this is the mutational variance. If we have n variants in Z, so Z is Z1 and Z2, we might imagine a mutational variance for one of the traits, mutational variance for the other trait, and there might be covariances. Now, if there's no interaction between mutation at these two different traits, these would be 0, and this is just the mutational variance, the input of mutational variance due to somatic mutation. To think about phenotype, we're going to go back to that idea of rho, this association of this environmental component in parents and their clonal offspring, but not their sexual offspring. So we make a vector of association parameters, ij, that gives the association between this environmental component in a stage I offspring and that same quantity in its stage j parent. And there should be ij's all over the place. But for a particular variable of this multivariate trait, we model it this way, all right? So basically, what rho tells you is how much of the offspring's environmental component is random. So if rho is 0, as it is for sexual offspring, this term is gone. And then you're basically just picking the environmental component from a random distribution. If rho were to be 1, this is 0, and the environmental component in the offspring exactly looks like the environmental component in the parent. And so this is just sort of the scale of where you are along that continuum. Let's do that. Yeah? So doing that, we end up having a probability density function for the phenotype of the clonal offspring. And all I really want you to get out of this is that we're going to use this matrix R. Actually, there should be ij's as well as the k's, but that's fine. And it's basically the identity matrix multiplied by the vector of these rows, and so these rows just appear in the diagonal. And so R in multiple, for a multivariate trait is this matrix, and later I'll show you a really simple example where we only have one trait and then it's just row. But you can, wherever you see R, you can think of just being row, basically. So then we use these to develop recursions for the genotypic and the phenotypic means, the change in the genotypic mean and the change in the phenotypic mean. And then to get the closed form for these, that's where we do that assumption of the Gaussian distributions. And there's the answer. Yay! That took a lot. I know it doesn't look simple, but we thought it was actually pretty simple because the thing that we started from was much worse. So we're like, okay, so what does this mean? What does this look like? It's actually, we can sort of break it down a little bit. First of all, you know what these things are here because I talked about them. Did I talk about them today? Yeah, I wrote them over here. They're gone. But so remember P, the matrix of phenotypic variances in the diagonal and phenotypic covariances in the off diagonals. And G was the additive genetic covariance matrix that had the additive genetic variances in the diagonal and then the additive genetic covariances in the off diagonal. That's what these are. The subscript just says it's for a particular stage. So we imagine each stage might have a different matrix. So that's what those two things are. These things are just functions of the entries, the mean entries of the transition matrix. And they're the means, again, taken over the genotype, phenotype distributions. So just as an example, this Dij is just the relative contribution of J to the I's, for example. Okay, and then this is the gradient operator. It's basically, you know, the dependency of the entries of the transition matrix, or whatever we're looking at, on the phenotype. We'll use that to measure selection. In fact, I think I have a note to say something at this point. Well, so we have some terms here that look like, for example, this term here, right? That term there. So if we think about things like that, that appear in the equation. This should be a subscript. For a single trait, so this is just that Z is just Z1, say something like that. That's just this. This is the and also in that we have J for Cij bar. And again, for a, should be a bar there. For a single trait, it looks something like this. Why do we care about that? We care about that because this is where we can measure how selection might act. Remember that these are the entries of the transition matrix, right? So they determine the growth rate of the population. So this measures how that transition matrix, and thus also how that growth rate, depends on the phenotype. And then when we give it, we'll give it a specific example later, but I wanted to write it up to sort of remind me to point to that later on. Okay, yes? Yes. Genotype as well? We're keeping track of both of them because when you probably could formulate this entirely with phenotype, but we track genotype so that we can see how the sexual reproduction is affecting it differently. You see it, I think it's more clear when I do a very specific example. But yeah, obviously they're related to each other, right? So you can sort of subtract them one from the other. I know. There's reasons, though. There's reasons. So if we just look at phenotype, let's look at phenotype first. Notice that R, so R is this, again, this matrix of those association. And what R does is it separates out the contribution of clonality. So here we have this is the relative, see, this is the relative amount of clonal reproduction, and this is the specific entry, Cij. It divides the contributions of the clonal parts of the transition matrix into parts that depend only on phenotype. So here's mean phenotype, and here's the phenotype covariance matrix. So mean phenotype and the variance in phenotype. And notice that then we also have, oops, wow, okay. That we similarly have terms for the transitions. Remember the transitions where I said the transition just carries its phenotype with it? We have these transition terms that also are multiplied by just mean phenotype or the variance in phenotype, right? So it's saying that you can take clonal reproduction and break it into two pieces. A piece that's like transitions, and then a piece here, this is the identity matrix minus this row matrix, that has to do with either the mean genotype or the variance covariance matrix for genotype. And that's like these are fecundities, this is sexual reproduction. So the way we've set this model up is we've taken clonal reproduction and broken it into two pieces. A piece that's like transition and a piece that's like sexual reproduction. At least for phenotype. Now for genotype, you know, we just have 2 bits. How does the genotype change? Well, there's just movement from J to I that carry the genotype, and those are the movements in the matrix. And then this part here is the combined effects of basically selection, like this here. So here we have the effect, actually this should look, so this is the, if this was a single trait, right? This is the additive genetic covariance or variance, and this is d log of A over dz, right? So it's how the log of those entries depend on the phenotype, right? So we have a non-selective and sort of selective bit. Yeah. Yeah. Yes. If we had no clonal reproduction whatsoever, this is exactly the same. But this is very different. So the phenotype is what's changing. The effect of clonal reproduction is changing how phenotype is going to track the change. So we'll see that in some really simple examples. Even if you don't have change, yeah. Yeah, it would still be more. Yes. Stage structure. Yeah. Yeah. So both stage structure and clonal reproduction are making this sort of ugly, but I know. I feel like people who are like a background in physics are like, oh, no. But biology is messy. Okay, but now we make it simple. This is what Daniel always, they keep saying toy model. Even there, it's not so simple. Simplest thing that we could think of is first we started with no stage structure, and just each generation, there's non-overlapping generations. Each generation, in the same generation, you survive, and then you reproduce if you survive, right? So we have probability of surviving, and then this sort of two reproductive things. So we can't even write a matrix. There's no matrix for this thing, just a single thing. So we have clonal and sexual reproduction. And now we say, okay, what if there is selection on one of the entries? Well, there's no entry of the transition matrix. One of the features of the life history, the survival probability, just for that case. So we just assume that it's stabilizing selection. And we have see, I promised we would see this again. We have some optimum theta. And we have the mean of this trait in Z1 bar. There's only one here, so there's really no need for that one, but that's okay. And so it falls off, right? So if you're right at the optimum, you're at some value, and otherwise it falls off. And we're going to track the amount of clonal reproduction as a relative amount. So just the amount of clonal reproduction as the fraction of total reproduction, just so we only have one thing to follow. So you can write the transitions for phenotype and genotype, and you see that, well, one thing you would see, if there's so clonality enters in as a joint parameter, R sub C times rho. So the amount of clonal reproduction times this matching, this phenotypic matching. If that's 0, then you get a much simpler looking equation. The equilibrium values for both of these, they go to theta. So adding clonality doesn't change the final equilibrium, but what it changes is the dynamics, and quite a bit. So well, because this is just tracking the mean, the shift in the mean. Yeah, no, this is it. This is mean. Oh, so you're asking where is sigma z here? So it doesn't enter into this. Yeah, I mean that's here. Okay, I should have. So remember, this was the phenotype genotype matrix before. So sorry, I should have said something that I didn't say. I dropped to one single z, and so now P, which was the matrix, right? P was Z1 squared, blah, 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 and the covariances, right? Now P is just, and G is just, yes, it's there. It just has a funny, yeah, sorry. I should have defined that. So we left it as P and G rather than writing it in the other notation, but now the matrix has become one entry, right? There's no covariance because there's only one trait. So that's where it's sitting. It's sitting. That's the phenotype variance, and that's the genotype variance. Okay, so to look at this, I thought one way to do it is to just numerically iterate these equations to sort of look at it. So I have a little sort of toy example. So this is the same. And we're going to make just a simple comparison between a case where R sub C times rho is 0, no clonality, and a case where it has an intermediate value of 0.5. Here is the mean genotype or mean phenotype over time. And here's the, well, it says adults, but there's only one type of organism, the population size, again, over time. And for no clonality, genotype and phenotype go together. It's this dotted line. And you see, oh, and for this numerical iteration, we started with z bar at 0, and theta is 3. And you see, it just does what it should do, and it goes to 3. This is deterministic, so there's no stochasticity, and it behaves nicely. And with clonality, the phenotype kind of takes off, but eventually, it does go to 3 eventually, and the phenotype does, but notice that after a little bit, they're both lagging. So you'd have to blow this up a little bit, but the no clonality case reaches that equilibrium faster. Evolution is faster. And I think that's sort of, sort of makes sense to us, right? That evolution is working more efficiently on these sexually produced individuals. But something interesting happens. So here's the population size. This is the case of no clonality. So, you start with some number, and we made it so it had to cap, so it didn't just grow exponentially. But it starts at some number, and, initially, because it's so maladaptive, it's really far off, the number goes down. And again, this is not a simulation, this is the iteration. Back up again. It goes back up again, even though it's, in real life, look at that, that's at 0, but it goes back up again. But with clonality, it doesn't go down as far, and it goes back faster. So this loss of individuals, which we said is very important for the ability of evolutionary rescue to happen, is dampened quite a bit by clonality. We called this a demographic advantage. So we have these individuals, if they happen to be okay, they make exact copies of themselves, and the population, makes it through. Now, we also looked at toy model number 2. This is the second less simple one. So it's almost as simple as before, but now we have 2 stages. Now you can move from this stage to the next, and you can stay in the same stage. And again, we have reproduction by sex and reproduction clonal. Now we actually do have a little tiny matrix to play with, it looks like. And we ask the same thing. Obviously, now we could have selection on the entire transition matrix, on a group of entries, on 2 entries, but you have to start somewhere. So we said, let's put selection again on the same thing we did before. Before it was just here, but it's the survival in the juveniles. It's the same form as before, Tmax. And we still follow the relative contribution of clonal reproduction. And now we have, we now have 4 things to follow because we have 2 stages. And it's actually, again, not so complicated. So P1 and G1 are these variances for the phenotype in stage 1, because that makes sense, that's where selection's happening. So that's where the variance there is introduced, and it has this shape. And again, they only appear together, R sub C and rho. If we do the same thing that we did with the other toy model and iterate it analytically, and sort of look at the same thing, now we're looking at changing that amount of adult survival. We're basically asking, how much does stage structure change things? So, so here's a little bit of stage structure, a little bit of surviving to here, of staying here. This is T22, of surviving and staying in this stage, adult stage. Here's a little bit more and a little bit more. It's the same thing where we started it at 0 and the optimum's at 3. They all go to 3. They just take a long time to get there, but they get there. And as you add more and more adult survival, it's slower and slower and slower. This should make some sense. Remember, selection is on the juveniles, right? So the more adults that are surviving, the more individuals that are sitting around, and selection doesn't have a chance to act on those. They're past that. So it's really slowing this down. But again, we see this demographic advantage. So here's this, the population size of the adults. This is for a little tiny bit of a smaller amount of adult survivor, goes quite down and up again, and then like this and like this. So it's again making it more shallow and making it come up faster. Now, it's important that there was remember we talked about trade-offs. We're not making a trade-off. So it's actually not surprising that we have this advantage to more adult survival because we're making things better, right? You can make this effect go away if during that shift, the adult survival increase is compensated by decrease in something else. So when I change the matrix to increase adult survival, I decrease something else. So this is a case, here's one example. I don't remember what we did. I don't remember which parameter we changed in this one. So we decreased Tmax. Okay, that's what we did. So we decreased some of the juvenile survival, some aspect of the maximum juvenile survival, as adult survival increased to equilibrate lambda to the same amount, and then the advantage goes away. So in this panel is what I showed before. So this is with no compensation, with no trade-off, and more adult survival is better. But if you make a trade-off, then it's worse, right? So obviously, you can make any advantage go away if there's an implicit trade-off. And so the question is there really going to be one? Not always clear if there is. So we see some evidence that there's not. So for the analytical results, we see that clonality and adult survival, which leads to stage structure, both of them slow the approach to the equilibrium phenotype. So they slow down the ability of the population to track that change in the optimum. But they both also reduce both the extent and the duration of that population size decrease. So they give a demographic advantage. But that demographic advantage makes you immediately wonder what happens if you add stochasticity. And so to do that, to think about that, what we did is individual-based simulations. To add stochasticity both of just sort of numbers, but we also add mutation and so randomness, environmental randomness as well. So I'm going to show you some results. We took the approach of Berger and Lynch's paper, did this sort of thing. I'm going to show you results for a single polygenic trait. So there's just one, instead of Z being the vector, Z1, Z2, Z3, there's just Z1. The simulation results that I'm going to show you have 10 loci with additive allelic effects. And I want to stress here that they're additive both within a locus, so there's no dominance, and across loci, so there's no epistasis. But I'm going to come back to epistasis in a minute. So I'll come back to that. The number of loci, we played around with it. It depends if we scale things differently. We could have more loci or fewer loci. That it didn't seem to matter as long as everything else was scaled. This is the distribution of those environmental effects. There's two mutation rates, one sort of gametic for sexual reproduction and one for somatic mutations. Again, this is the relative amount of clonal reproduction, this association parameter. And then we looked at two different kinds of phenotype change. A one-step change and continuous change that was linear. So we looked at those two options. Let me go back to this thing about epistasis. There's no epistasis built into the loci, but I said, we have well, pretend that's a Gaussian. If you have a Gaussian fitness function, as we've talked, in fact, we talked about it just today, there's having curvature in the fitness function immediately introduces epistasis for fitness, right? So, and you can imagine that small jumps, right, will tend to have maybe, sort of positive epistasis, where they increase the effects, positive effects of fitness, and we might imagine big jumps maybe make you miss. So, I mean, it depends on the details of exactly how you're allowing the mutations to change phenotype, but when I say there's no epistasis, it means we just didn't put epistasis in for the loci, but as soon as you have curvature in your fitness surface, there's sort of epistasis is a result of that. Okay, now sorry, the diagrams are a little complicated. But again, no stage structure and the simple stage structure case. This is a one-step change in the phenotype. So we start at phenotype something, and we move the phenotype to theta. And so down here is how big that jump is, the change in theta. And then what's given on the vertical axis is the probability of persistence. I'm trying to remember how many simulations each one is. Some large number. Um, 400 simulations, so it's the proportion of 400 runs where the population persisted to some time that we determined. And this is keeping rho constant but changing clonal reproduction. The red arrows show increasing clonal reproduction. This is keeping the amount of clonal reproduction constant and changing that association parameter. The red arrows show increasing rho. And just to orient ourselves, if we make the change in the change in theta big enough, everyone goes extinct. The probability of persistence is 0. That makes sense. If it's a really, really big change, there's no way to survive. If the change is small, everybody's fine. They make it. It's all right. Just a little change. It's in the middle, right, that there's some difference. And so what we see is as we increase clonal reproduction, or as we increase the association parameter, we increase the probability of persistence for a given change. In other words, clonal reproduction and this association in E makes things better. It makes the populations more likely to survive. That's true here as well. And the other thing you might notice is that compared to this set of curves, these guys are shifted slightly to the right. These are also a little bit shifted to the right. And generally, stage structure also makes things a little bit better as well. So stage, and it's because of that demographic advantage that we saw. All right, so they're making things better. We can lose that demographic advantage of increased adult survival if, again, we make that compensatory change. So if maybe we decrease fecundity with the increased adult survival, then we see that adult survival, increasing that decreases probability of persistence, and it makes it worse, the more clonality. So obviously, if there's a trade-off, we can lose the advantage of adult survival. But nevertheless, greater population persistence with more clonal reproduction and higher environmental component association. And if you think about it, it sort of makes sense. This is a single change that occurs in the optimum. And even though there's mutation in these simulations, for a single change, they've got to basically, that population, if it's going to evolve, it's going to evolve from the variation that's existing at the moment that the change happens, the standing genotypic variation. And so if you think about clonal reproduction, if you happen to have some genotypes in the population that can make it through that selective event, clonal reproduction lets you make exact copies. And rho lets you, if rho is high enough, lets you make exact phenotypic copies. So if the change is not too big, and you have enough standing genotypic variation, you can sort of make it through this. But we see something completely different when we have continuous change. Oh, and this is, sorry, I should have said this. Stage structure also increases the probability of population persistence, and it's that demographic advantage of the adult surviving, basically. We saw in the analytical case as well. So, even more complicated, but that's okay. So now we have a continuous change in the optimum. So now we don't just change it one time, but we change it every single generation, or every time step, rather. And so this is the rate at which the change in theta happens per time step, increasing this way. And now this is sort of a split diagram. The top part is the probability of persistence, and the bottom is an unscaled measure of lag, of evolutionary lag. It's how far from the optimum the population mean currently is. And the way we did this so that down is bad no matter what. So here's probability of persistence 1 is good. You always persist. 0 is bad. And then this becomes more and more negative when things get worse. So down is more and more lag. You're further down the slope, if you like. So now, if you look at both the no stage structure case and the stage structured case, what you see is increasing clonal reproduction or increasing the association parameter, which is the direction of the arrows again, now makes things worse. Now, you know, for a given rate of change, the more clonality you have or the more association you have, the less likely the population is to persist and the bigger the lag. So it's not as good. And it's a little hard to see in this scaling, but remember before the lines were shifted to the right, now they're shifted to the left with stage structure. So age structure also makes things worse. Does that make sense? Yeah? So with a continuous sort of linear change in the optimum phenotype, we see decreased persistence and more lag with more clonal reproduction in higher row. And again, this is one of those things where the model, when we first did this, we sort of had an intuition, and then we were wrong, but then afterwards it seems so obvious why we were wrong. It's like, why didn't I think of this before? If you think about what's different about this type of simulation, if you continually are changing the optimum, you rapidly use up the standing genetic variation. And, you know, we could think about the additive genetic variance that we wrote out for, you know, multiple loci. And you know, this is one of the things where I can't ever remember PI, QI, stuff squared that I don't remember. There were terms. And as you apply selection to this additive genetic variance, you're fixing some of these alleles. You're losing genetic variation, and soon you sort of run out. So this type of selection depends on de novo genotypic variation. So in these simulations, we might ask ourselves, where's that de novo genotypic variation coming from? It's coming from three sources. The first source is mutation, which is happening. The second source is recombination, which is, of course, happening in the sexual offspring and not in the clonal offspring. And the third source of randomness is that random environmental, part of the environmental component that was random. And when we increase rho, we decrease that part of that random environmental component. So a lot of the new genotypic variation is decreased in both clonal and with the matching. And stage structure has a really interesting effect. It decreased persistence and increased lag. Stage structure, I could write out, we worked out an equation for the generation time for stage structured populations. And if you increase adult survival, you increase the generation time. When we define it the way we did the other day in class, where we defined it as a meiotic generation time, the time from stage 1 to stage 1. And remember we said that for stage structured, that's not a constant, but rather a distribution. But nevertheless, that distribution's shifting when we add more adult survival. So we're increasing generation time. Well, if we increase generation time, each generation, we have more steps of environmental change, right? Because we have continuous. So more environmental change. Another thing is the way we model this, phenotypic selection is happening on juveniles. If we have, if the population's near the carrying capacity, which, except for some of these things that were really going extinct, they were at least initially, then the proportion of the population that is juveniles, that can actually be under selection, is small compared to the proportion of the population that's adults, right? So we have more adults. So it basically decreases the effective population size of the component of the population that can undergo selection. So it makes selection less efficient. And finally, I did not come up with this term. One of my co-authors did. I'm not very poetic. He said, it's like gene flow through time. And I sort of saw what he was saying. This is Bob Holt. If you ever meet him, you'll know that it was him. So it's sort of this maladaptive gene flow through time. Let's think about this case where the optimum is shifting and shifting and shifting and selection's acting. But selection's acting on juveniles, on the young individuals. If we have a lot of adults who survived, when did selection act on them? A long time ago. And so they have a phenotype that is maladaptive to the current environment, to the current theta. So it's a kind of gene flow through time that's not good, right? So they're coming from the past. And I could make a joke about this is why you don't let old people vote, but I won't. See, I knew Magnus would laugh because we were talking about this. So we have these issues of stage structure making things worse with this continuing change. Okay, so how will these types of organisms respond to environmental change? Like all, I think, interesting questions in biology, the answer is it depends. But what does it depend on? That's interesting. Obviously, the scale of change. Does the population experience that change as a single transition or as a series of transitions? We can ask about, this is a scale question, right? A scale of environmental change time versus generation time and mutation time and things like that. And so for lots of the processes that we often think of, maybe environmental change is very slow compared to the experience of the organisms. But for things like, well, certainly climate change, but I was also thinking of organisms that find themselves in new environments, invasive organisms, the scale of change can be very fast. But that's also going to depend, on the generation time of the organism. So we can think of organisms that have very long generation times versus organisms that have short generations. This is rho, basically, the amount of phenotypic matching between organisms and their clonal offspring. That's going to depend on sort of two things. One thing that we kind of didn't consider very much in the simulations, although it came out in the analytical work, and that's how much dominance and epistatic interactions affect this phenotype. If they affect it a lot, rho can be rather high. The other thing is the kind of clonal reproduction. So if it's something like maybe those hydra or vegetative spread, we expect rho to be quite high. And if it's a different kind of clonal reproduction, it might be low. So this might depend on the type of clonal reproduction. And finally, the existence of stage structure of long-lived organisms. I had a note that I wanted to say something. Oh, yeah, so again, we have this increase in survivorship and, of the adult stages. That can provide this demographic advantage that we saw. Remember that this somewhat cartoony view of what happens under a sudden change in the environment and whether evolutionary rescue. Having adult and stage structure, adult survivorship and stage structure can give you a little bit of a demographic advantage so that when you dip down here, you don't dip as low, and maybe there's more of a chance for evolutionary rescue. But at the same time, it can weaken if most of selection happens early in the life history, it can weaken the force of selection. So I guess that's another question you have to ask yourself for this organism and for this phenotype. Where does selection happen? For a lot of organisms, we maybe do think that juvenile survivorship is a strong selective force. I was talking to somebody who works on corals, which of course match this kind of life history. And for corals, a vanishingly small proportion of larvae settle and become adults. So obviously there's there could be very strong selection on that point. It could be totally random and then it doesn't matter. Okay. And that, I finished early again. See, I have no ability to time this. But I do want to say that this work was done with Bob Holt and Michael Barfield, who are at the University of Florida, and then Carlos Fernandez, who was an undergraduate in math, who worked in my lab while he was an undergraduate at KU. And funding from NSF and funding from various things at Kansas, which I always feel like I should thank because soon there will be no more funding, and this will be like a historical remnant of the olden days. Okay, and that's all I had. Thank you. But I'd be happy to answer questions. And I know you guys have questions. I know you do, because it wasn't that clear. I don't think that was that clear of a presentation. So. Yes, Magnus, good example. Yeah. Yeah. Yes. I think naively, and in this case I am very naive. Naively, I think that the kind of selection that I'm sort of interested in, this sort of climatic change and invasive populations, that selection, the strength of selection is pretty, so we're in the strong selection, and I don't know if we're in weak mutation or not, but we're definitely in strong selection rather than very, very weak selection. So I was just thinking of Jacquem's question of, do you move, do mutations have a chance to fix, or are you doing an adaptive walk? And I think in this kind of thing, selection is strong enough that you're probably moving around, you know. Whether the directionality is, that's a good question. Right. It's, and, right. Yeah, and we did allow new mutations to happen in the simulations, but I am, there's something about the simulations I didn't say, which I think is important. So we start the simulations and we run them for some number of generations and then we let them reach sort of mutation balance. You know, we let them get to a certain amount of, so there's plenty of, we start with a population with plenty of genetic variation. And we're sort of assuming that, you know, that might be a similar state to lots of traits. And so I think almost all of the de novo genotypic variation is not coming from the mutations in this. This is a short time scale. From the simulation. It's actually coming from recombination. It's coming from a sec. And I have I think that this model really applies to shorter time scales. De novo genotypic variation. I was careful to say genotype because it's not de novo, it's not new to the population on an allelic level, but it's a new genotype. So it's new genotypes. So how can we get new genotypes? So I didn't mention, and I should mention for the students, there is a huge and very interesting literature on evolutionary rescue, which I did not review in any way. And a lot of those models are looking at a few different things that I think, well, one thing I think is really interesting, that I didn't touch on at all, is the genetic architecture of whatever the trait is that allows you to make it through. And things like epistasis, but also whether you have alleles of strong effect or lots of alleles of small effect, and what the mutation rate is like. And so, yeah, this model really glossed over all of that and just asked this, it's very simple, I think it's a very simple question of sort of how does this just one aspect of the life history change how likely you are to persist if there's a change. But I think that the biggest question is scale. You know, what is the scale? Is it fast? Is it one change? How does the organism experience that change, and how is that scale relative to how fast selection acts? Obviously, the strength of selection could be very strong, but the speed of selection doesn't just depend on the strength of selection, right? It depends on other things as well. So you know, I don't know, but I think that's an important question. But certainly, you can get some intuition from asking what happens at slow timescales versus fast timescales and things like that. So I think there's plenty of room to ask that type of question. I think that's a good set of questions. Yeah. Well, we got the analytical result that there's a demographic advantage under a change. We got without making an assumption about the distribution. In fact, I think you could assume that we just assumed a large number of genetic loci, and you know, we did assume a Gaussian distribution of genotypic Gaussian. I'm trying to remember how many different combinations, but you know, that's the problem with simulations. I mean, they're fun and they're great, but there's so many different degrees of freedom that, you know. So one of the things we didn't explore, I think we explored just changing the number of loci, and you know, it didn't make a big difference, but that's not too surprising because 10 is actually, for quantitative genetics, 10 is a lot. So it's like 10 or 100, doesn't make a difference. But we didn't look at less than 10. So I think we didn't look like at a major locus and the effect, because then we worked in sort of the world of a smooth distribution of phenotypes and things. So we didn't look at that kind of thing. And we also didn't look at, though all of the loci have equal effect. They have different effects because they're chosen from a distribution, but we don't have like some loci of major effect and loci of minor effect, which I think that could change a lot of things. So we did not explore the genetic architecture, I think, probably at all. So, and I think that's important. I mean, we kind of took what's sometimes considered sort of a standard quantitative genetics to have enough loci that are independent to get a nice distribution, and didn't worry about it further. Yeah, we did. So, but in the simulations, that's not true. Because in the simulations, we didn't make any assumptions. Oh, that's good. This brings me back to something I said I would come back to. So one of the things the reviewers, when this got published, said is, hey, you're making assumptions of Gaussians for genotype, and but that's, you have with clonality. So what we did is in the simulations, there's no assumption of the distribution of genotype and phenotype. That comes out naturally from 10 loci, and sex, basically. That's a good question. Because I don't remember that I did that. I do. Well, I expect it should. In the continuous case, it should, especially, right? Because especially in the cases where those things are well, at least, sigma g definitely is decreasing, right? Because that's the whole thing. It's losing its ability. But I don't think we looked at, so I don't know, oops, sorry, I don't mean to yell. I don't remember that we graphed the change in the variance with time. In the simulations, we didn't assume anything about the variance. We just let it go, and we did set up the simulation so it built up a certain amount of variance at the beginning, so it had variance. But yeah, I don't think I looked at that. So that's a good question. We could easily, we have all that stuff, so we could look at it. But it's a good question. In fact, I wrote it down. I will. Yes. Oh, no. You're going to have to use the fancy microphone. Did we lose all our microphones? This is like on those talk shows where you ask a question. But this isn't Oprah. You're not all going to get a car. That's an American joke. That's not funny. Okay, go ahead.
So following up on Magnus's question, I think you're not really considering fluctuating selection, right?
No, we're not. We're actually doing that now. But for this paper, we didn't do it. I think we had sort of in our mind, to be honest, when we were writing this, we were very obsessed with an invasion or something that's a sudden change, like a strong shift. Because we were talking to these people who work on all these cool clonal seaweeds, and they come in and they grow all over the place, and we were like, what is going on? So but I think that's a good question. We're sort of the problem. It's not a problem, but one of the things about fluctuating is then how do you fluctuate? Do you fluctuate regularly? Do you fluctuate irregularly? And there's all the period of the fluctuations, and there's a lot more there's a lot more degrees of change. And we did actually want to try to publish it at some point. It was already ridiculously long. But that is actually what we're looking at. It's one of the things we're looking at. That, and then we're also considering spatial structure, which is completely missing from all of this, and that's the other thing we're considering right now. But yeah, no. And that's, I think given our conversation earlier about, you're moving on a fitness landscape and the optimum changes, but it could change again, right? And then you could be doing this walk all over the place. I think that's a really important question. Because this also assumes, right, that this is directional selection. Theta is changing and it's just going that way. How likely is that to happen for a long time? I don't think any of us have a problem with theta, with the optimum changing and continuing to change. But is it likely to change in only one direction? I don't know. But that's a really good question. I'm good. Oh, more? Okay, just because I'm standing here.
No, so
yeah, that's baked into the transition matrix. So the idea is that you would, and one of the things that I thought somebody would ask me was, wait, if you have a clone that's spreading vegetatively, when does it become a new, transition? When does it become two things and not just one with a runner? And I've actually thought about that a lot. I've published on that stuff, and I thought about it a lot, and my earlier models talked about it. And I've always taken the viewpoint that I think people who work with age-structured and stage-structured models often do, that when they're physiologically separated, so one piece can die while the other piece lives, they're two things. So if they're still physiologically connected and the death of one would lead to the death of the other, like the hydra, if they're still budded together, that's not true yet. But yeah, that's baked into the, we don't have the transition matrix. The transition matrix those movements are different from sex. And they have very different probabilities of survivorship. They're often much higher, right? Because a seed, you know, it's very low probability of establishing. Yeah. Yep. What's not into that, and this is where I said we were thinking about space, is the models I'm working on right now, which have to be done if I give a talk in Montpellier, but maybe I won't give a talk in Montpellier, but that's what I'm hoping to do, is to think about the fact that if you're thinking about spatial structure and migration, sexual offspring often move very differently, than clonal offspring, and the plant is a good idea. You know, so the sigma that is the spread of the variance in the distance between me and my offspring is probably quite smaller for vegetative space, much smaller for vegetative than it is for sexual. So it creates this correlation of environment with clonal offspring that is somewhat broken with sexual offspring that end up going further away. So there's some interesting aspects to that we're thinking about.